And he is the science area lead for computing, analytics, and modeling at the Environmental Molecular Sciences Lab. He got his PhD from MIT in electrical engineering and was also part of the Department of Energy's Computational Science Graduate Fellowship, which is how I met him, since I'm also currently part of that fellowship. Since then, he has also worked at Argonne National Lab, Rush University Medical Center, Northeastern University, and GlaxoSmithKline. Today, he'll be talking about opportunities at United States National Labs for bioinformatics and general career advice. So if you can all join me in welcoming Dr. Bardhan.
Thank you very much for that kind introduction, Olivia, and thank you all for having me here today to share with you some of the cool things that are happening in and around what we call EMSL, the Environmental Molecular Sciences Lab. So in order to sort of set the stage for what EMSL is all about, what it does and why it does what it does and how it goes about it, I wanted to start by zooming in from sort of a high-level view of the U.S. Department of Energy and its Office of Science, introduce the National Lab System and PNNL within that system, EMSL within PNNL, and then I can talk a little bit about EMSL's user program. It's a scientific user facility, which means that PIs can apply for time on our instruments, whether those are computers or our staff can operate some of our high-end instrumentation. And that creates opportunities for collaboration for students, postdocs, and faculty to visit the lab. And I'll talk about how to get engaged with that user program. I'll share a few thoughts on careers at PNNL and EMSL, some broader career perspectives from, as Olivia highlighted, I have had kind of a Brownian motion throughout my career and some thoughts on that. And then I'll close with some more information or ways where you can go to learn more. All right, so the U.S. Department of Energy is a gigantic organization. It has a large number of different kinds of offices, but the key ones here are the program offices, the things that do things, and the Office of Science is just one of those. But the Office of Science itself is the nation's largest federal sponsor of basic research in the physical sciences. And it has been that way for many, many years. Importantly, the U.S. Department of Energy's Office of Science sponsors research at universities. People can apply for grants the same as with NIH or NSF. But also the U.S. Department of Energy operates through contracts a large number of national laboratories and other kinds of research institutions. They also maintain scientific user facilities, of which EMSL is one, and I'll describe a little bit more, which are available to the international science community. And the overall goal of the mission of the Office of Science is to advance the energy, economic, and national security of the United States. So an org chart is just one way to explain the bewildering complexity of the Office of Science. The director of Office of Science is Professor Asmarat Asafal-Berry, who's a soil scientist from UC Merced. She's an extraordinarily accomplished scientist, and we are very, very fortunate to have her as our current director of the Office of Science. There are 6 program offices that are our science programs. Those are shown on the left. Advanced Scientific Computing Research funds a variety of activities in applied scientific computing, applied math, and so forth. And BER, the Program Office for Biological and Environmental Research, is the one that supports and sponsors EMSL. Within BER, looking down another length scale, if you will, we have 2 divisions. One is on biological system science, whose primary activities are in genomic sciences and bioimaging. And the other half of the house is the Earth and Environmental System Sciences Division, whose activities include atmospheric research, environmental system science, and earth and environmental systems modeling. So if you've heard about climate modeling over the last few years, especially, that is a lot of that leadership comes from BER's Division of Earth and Environmental Systems Science. Each of these divisions also operates key scientific facilities and infrastructure. On the biological systems side, there's the Joint Genome Institute, which is located in California, and a variety of infrastructure supporting structural biology at various, for instance, light sources around the country. On the EESSD side, there are two user facilities. One is the Atmospheric Radiation Measurement, or ARM facility, which is operated by PNNL, and the EMSL user facility, also operated by PNNL. So zooming in to the National Labs system as a whole, the DOE operates 17 national laboratories, which are distributed across the country. There are 10 Office of Science laboratories who serve the broad program offices shown in the org chart. There are 4 DOE laboratories that have a variety of other functions and sponsors within DOE, for instance, related to renewable energy. And there are 3 National Nuclear Security Administration laboratories. Those are the ones in blue. PNNL is one of the Office of Science laboratories and has 3 key areas of focus. One is in the broad spectrum of scientific discovery. One is in specific areas related to sustainable energy, and a third in national security. One of the things to think about, to be aware of, is that the national labs are not operated by the Department of Energy itself. Rather, they're managed and operated by either consortium or private institutions that manage and operate the labs on behalf of the Department of Energy. And PNNL is managed and operated by Battelle. Within the 3 main themes of scientific discovery, sustainable energy, and national security, I'm going to talk about the scientific discovery elements, and particularly related to biology, earth and coastal sciences, and chemistry. The Office of Biological and Environmental Research runs a number of large-scale, extended in duration research efforts, which they call science focus areas. The ones that are centered at PNNL have significant bioinformatics elements. These aren't my specific area of interest. They do intersect with the EMSL's activities in certain ways, so I wanted to highlight them as opportunities and to encourage you to reach out to me if you'd like an introduction, or these are very friendly folks. You should feel free to reach out to them on your own if you like. One is in soil microbiome science. The PI is Kirsten Hofmackel, which is looking a significant amount at how soil moisture changes the composition and activities of soil microbiomes. We have a new science focus area led by Rob Egbert on engineering functions in soil microbiomes to ensure that they don't, if we're engineering microbiomes, that they don't get out of control or migrate, sort of the Jurassic Park problem. And a third at the environmental scale is led by Tim Scheibe on river corridor hydrobiogeochemistry. And so that encompasses everything from the microbes in the river corridor to the ways that soil and materials are transformed chemically by the activities of the microbes. I've put links where possible at the bottom of all of these slides, but if there come things where you don't see those links and you'd like them, please don't hesitate to reach out or ask at the end of the presentation. A fun digression, I have some very nifty colleagues. Those of you who spend time online may have seen comics by Red Pen, Black Pen. The artist is a microbiologist and bioinformatician named Jason McDermott, who is awesome. This is his activity in his private time. It is not part of his work for PNNL, but it is a way that he expresses himself and enjoys connecting with colleagues. His actual scientific research connects microbiome composition and functionality. It uses multiple types of omics data from metagenomics to metatranscriptomics, metaproteomics. He also uses a lot of high performance computing and works with experts in graph algorithms and AI ML to develop faster, more accurate techniques, for instance, to understand relationships, evolutionary relationships in protein families. All right, zooming in a little bit more now to EMSL itself. EMSL is a scientific user facility. That means that we make our capabilities, our instrumentation, and our expertise available to scientists around the world, subject to the PI writes a proposal that is aligned to the interests of, aligned to the mission of the Biological and Environmental Research Program Office. It is within the capabilities of our instrumentation. And it reviews well according to external peer review panels that we convene after calls for proposals close. It's named after Bill Wiley, who became the director of PNNL in 1984 and was a visionary leader. In response to a widely publicized report about the potential of integrated research capabilities, he proposed in 1985 a center that would include environmental and modeling and biological experimentation capabilities. And it took 12 years before that dream became a reality. And unfortunately, he passed away in 1996, just a few months before EMSL was dedicated and about 1 year before EMSL opened for business. So on October 1 of this year, just a few weeks away, EMSL will celebrate its 25th anniversary of opening its doors for operations. And that is tremendously exciting, and it is thanks to a decade plus long vision, drive, and determination to see this kind of integrated capability brought to the scientific community. We just want to look at some of the numbers about how much happens at EMSL. Over the last fiscal year, this is just through the 3 quarters of fiscal year, which of course runs awkwardly October 1 through September 30th. We had 239 active user projects, means 239 funded proposals. And across those projects, there were 688 different users, people who were part of proposals. 19% of those this year were new to EMSL. They'd never had a proposal funded or awarded by us before, yielding 288 publications with an average impact factor of 8.9. And 44% of those user publications include the EMSL staff involved as co-authors, indicating the degree of scientific contributions beyond operating the instruments, but actually providing key subject matter insight into protocols, into techniques, and into experimental design. For the last large number of years, EMSL has organized around 2 science areas. One key one was environmental transformations and interactions, the E in BER. The other was functional and systems biology, the B in BER. We had a new director take the office in a few years ago, I believe in 2019, and Douglas Manns highlighted that the importance of computing and the ways that computing integrates across biology and the environment, and the growing specialization of techniques in informatics and modeling and simulation meant that EMSL should create a 3rd science area specifically focused on computing, analytics, and modeling. And so that's the role that I have, and it is a tremendous privilege to support the breadth of ongoing research in the other 2 science areas, as well as to try and bring in new expertise, new techniques, and new perspectives on how to go about doing integrated science. Highlighted earlier that there are 2 halves to the BER house, and our organization in these 3 science areas really reflects that. Each of the 3 science areas has a particular strategic science objective, or SSO, because we like our acronyms. On the biology side, we're talking about creating a digital phenome, which could be at an organismal scale, a single cell scale, or it could be at a microbial community scale. And we could be talking about building that kind of digital phenome model based on any of a variety of kinds of data. On the environmental side, on the right half, we have a new science area lead, John Barger, who joined us from SSRL. He is looking at building a collaborative of observation sites around the continental United States called the Molecular Observation Network, where folks are taking standardized protocols for observations and soil cores, shipping those cores to EMSL for a standard suite of studies, whether that's X-ray computed tomography, a variety of organic matter analysis, and so forth. And each of those strategic science objectives has associated with it 3 what are called integrated research platforms. Think about these as being the tactical teams that execute particular capabilities and have an eye towards the evolution of those capabilities. We have a structural biology IRP, which focuses on things like cryo-EM, NMR, and so forth. We have a cell signaling and communications IRP, a biomolecular pathways IRP, which includes an awful lot in the realm of mass spec to understand proteomic data and take metabolomic data. On the environment side, there's biogeochemical transformations, ecosystem interfaces, and plant and ecosystem phenotyping. And in the middle of all of that, we have what we're calling systems modeling and what we will be creating as a data sciences IRP to serve the different needs of those 6 IRPs in the other science areas. So where we are today is a time of rapid growth in computing, because we only have sort of 1 IRP, but the vision is to have 3. And for that, we're going to need people who appreciate the subject matter of biology and environment, people who understand deeply the data types and all of the characteristics that come with those, whether that's noise or incompleteness, or all of the complicated context associated with particular kinds of species and communities. One element of Bill Wiley's vision that EMSL has led in since its inception is in high performance computing. If you've ever heard of Linux clusters being a major factor in high performance computing, some of the early work to set that in motion was done at EMSL. Our current supercomputer is called Tahoma. It's a heterogeneous supercomputer, which means that we have some nodes that are configured in one way and other nodes that are configured in another way. I'll provide a few details on the next slide. But we designed that on the basis of recognizing that this vast diversity of scientific applications isn't very well served by having all the nodes conform to a particular configuration. We also have, as you can imagine, from a lab with over 150 scientific instruments, we also have an extraordinary appetite for data storage and management of data over a very, very long life cycle. And we also need to capture metadata to ensure that the data that we take as part of taxpayer-funded research is made available to the scientific community according to FAIR principles. And we also are increasingly offering cloud computing capabilities such as Kubernetes, classic virtual machines. We recently deployed a way that lets you get onto our supercomputer just by logging in via the web using Ohio State's Open On Demand software platform. And the goal is to make sure that people are able to access the full power of this machine with as minimal knowledge as needed beyond the actual scientific expertise that you need for solving your problem. So as I said, Tahoma is a heterogeneous supercomputer. We have 180 odd nodes, which are 36 cores each and several hundred gigabytes of RAM and a lot of local flash storage. And we had 24 analysis nodes, each with multiple high-end GPUs and 1.5 terabytes of RAM. And the reason that that's very important, I presume some of you have experienced this, is that a lot of bioinformatics and assembly algorithms are extremely RAM hungry, and they don't split well across processors in the way that some of our friends who do computational fluid dynamics are able to. So the machine was explicitly designed with in mind the notion that some algorithms that are very, very well tested and that we understand need a lot of RAM for doing leading edge calculations and assemblies. One of the areas in which computing at EMSL is sort of distinct and I think very fun is that there's a unique combination of the resources, the actual hardware and software that we provide, and also expert support. Our operations team on the computing side has decades of experience in providing and provisioning software on HPC systems. And I just want to give a very quick example of that from a project that was analyzing metatranscriptomics data. So Emily Graham, who's now one of our IRP leads, was studying microbial communities in wetland soils with metatranscriptomics. And the real challenge here was that a lot of the transcriptomics analysis and assembly softwares are really not well designed for the scale of data taken in metatranscriptomics experiments. And so what they had was about 270 million RNA reads, and the analysis would take their staff months to complete an assembly. And even when it took months, they were required by the constraints of the machines that the computers that they were using to take certain approximations that they knew they wouldn't take if they had all the resources that they wanted. But with this specialized heterogeneous computer, they're now able to run a complete analysis in under 24 hours, thanks to the combination of a large number of fast nodes and a few nodes with enormous amounts of memory. And what made that possible was that the scientists, the domain experts, knew a certain piece of software almost did the trick. And it was our high performance computing folks who said, all right, I don't know that much about bioinformatics, but that's okay. I'm going to look at the libraries that you're calling. I'm going to look at the patterns of data usage, and we're going to be able to split the actual execution of the assembly into two parts. And then one can be parallelized very easily, and the other can be run on our big memory nodes. And so you can imagine that if your analysis goes from taking months to taking just under a day, that can actually change the way in which an experimental scientist approaches their field campaign. If they know that they can go take some data and have the analysis complete before they take the next round of data, the difference between months and days might be huge for understanding response to things like flooding. So with that example, now I'll talk about the user program more generally. We offer a wide variety of kinds of proposals. Some, like the limited scope, are very short in nature. The idea here is let's see if our kind of equipment, whether that's a particular mass spec instrument or our supercomputer Tahoma, is a good fit for the scientific needs that you have. You can submit a limited scope proposal any time of year. It just is only going to be a month long. We have exploratory research opportunities, which last 9-ish months, and the topics change year after year with changing BER priorities. We have two 2-year mechanisms. One is called large-scale EMSL research, and one is called FICUS, and I'll describe FICUS on the next slide. The important thing is that the exploratory, the large-scale, and the FICUS all go through an external peer review panel to ensure that the quality of research is high and that the alignment to BER interests is also high. All right, FICUS. So this is an incredible acronym: Facilities Integrating Collaborations for User Science. right? The idea is pretty simple, and it is a really, really cool one. What if we wanted to do science where part of the capabilities that we need are at one user facility, like EMSL for imaging of soil samples? And what if part of the capabilities that you need lie at another user facility, like the Joint Genome Institute, which has massive sequencing capabilities? Well, if you could only apply to each one separately, it might be hard to get both approved and then do your science. So why don't we have a joint system where we can review proposals together? This year, we have a ficus that involves EMSL and JGI. We also have ficus calls with NEON, the National Ecological Observatory Network, a separate one with the Biosands Beamline at Oak Ridge, and one with the Atmospheric Radiation Measurement Facility. We're also piloting other potential partners because we have a lot of user facilities that are operated by DOE where science could be enhanced by this kind of large-scale collaboration. All right, how does it actually happen that a PI gets access to all of the goodies of EMSL and its expertise? Well, it starts by EMSL opening a call for proposals. That only happens after the staff scientists and the leadership sort of have a huddle and talk about what capabilities we've recently brought online in the last year, what we're hearing from the Office of BER as to their emerging priorities, where are calls for proposals going to be coming from them, what we might know about that. And then we share our draft topics with BER, they provide some feedback, and then we open a call to the public with a specific set of call topics in certain science areas or with certain science capabilities. There's then a letter of intent process. The letter of intent is sort of 2 pages, and we look at it and understand, is this science aligned to the call topics? Is it plausible that the scope of an investigation could be compatible with the size of projects that we can actually support? Because these aren't $10 million projects that we can support, they're smaller than that, okay? But if your letter of intent, which is just 2 pages, seems to be aligned to the call topics and within the scope of an EMSL user project, we say, great, let's do this. And we encourage you to interact with either the science area leads, which are me, John Barger, or Scott Baker, or with the leaders of the IRPs whose resources you would be asking to use. And this way, you develop an understanding of precisely what is best as far as resource usage, according to their view and yours in collaboration, and also to decide on a scope. After you submit the proposal, which is just 4 pages, then there's some internal reviews to make sure that all the boxes were checked that need to be checked, and then it's sent out for external peer review, and we hold a panel just like with other funding agencies might. The top scoring proposals are we then look at the resource needs in more detail, and the peer review panel creates a ranked ordering of which proposals they recommend we fund, and we fund up to the line where we run out of budget for that year. Makes sense? And then we let the PIs know, and the project managers and the IRP leads coordinate kickoff meetings. So one advantage to working with EMSL is that your project, the experiments you want done, and the different kinds of analysis you need coordinated, those are managed by professional project managers employed by EMSL. We do much more than just run a large user proposal mechanism. We also want to grow the community and help the community build skills of importance to BER and to grow awareness in the community of our expertise and that of our network of collaborators. As part of that, we host an annual summer school. One of the positive things to come out of all of the terrible disruptions of COVID-19 is that EMSL was able to move these summer schools online. That allowed an order of magnitude more participants than had ever been possible before. So as shown here, 2020 focused on microbial dynamics modeling, and it was the organization of this was led by Tim Scheibe and his SFA key staff. As shown at the top inset, the videos for these summer schools are available on YouTube. I have a link at the end of the talk, but more practically, if you just type in EMSL Laboratory YouTube, our channel will come up. 2021 was using multiple forms of omics to understand and model biochemical pathways. And this summer's was on understanding soils. I want to highlight that this included participation from another Office of Science initiative called the National Microbiome Data Collaborative, which is coordinated through multiple national laboratories, including PNNL and Lawrence Berkeley Lab. I won't name all the participants, but my apologies to those I can't remember off the top of my head. All right, so I will say a few things about careers at PNNL and EMSL. The best place to start is just with careers.pnnl.gov. We are always looking for folks at a full-time level, postdoctoral positions, and we have a very active university internship program. If you go to the website and all of this that you're hearing from me sounds appealing, or you'd like to learn more, but you don't find any openings on the website, please reach out and email us. You can always email me at the address shown here, and I'm happy to connect you with potential hiring managers or folks who might know more about upcoming opportunities. One of the things that makes PNNL distinct, and I think a great place to work, is that it follows sort of a matrix organization principle, which is to say I have a line manager who is responsible for checking the boxes about my annual activities and HR functions and my annual performance reviews. But I don't work for that person. I rather work on projects that are run by different scientists or different staff members. That creates a lot of opportunities for you to have control over your career growth and development in directions that you think are important. So my line manager is a good human. I like him, I respect him, and he's a good line manager. He and I talk about how I'm spending my time, 20% on this project, 30% on this project. But in point of fact, I have multiple bosses because on that 20% project, I have one boss. On the 30% project, I have a different boss. And so if I wanted to get into, say, project management, or at least try on that role, I have the ability to go out and find a senior project manager and say, hi, Linda, I would really like to try this. It seems like a good fit to how I like to think and organize things and help people keep on schedule. I'd like to try it. And then that person might say, yeah, I have a sort of, assistant project manager opportunity coming up. So there's a lot of richness in your ability to sort of explore career tracks or grow new skills. I think that's great. One of the things about that, which ties these two bullets together, is that in fact, sometimes a PI on a research grant will have support for maybe a quarter of a postdoc, but they're not going to advertise for a quarter of a postdoc and then hire somebody and then not be able to fund them. So sometimes reaching out is really helpful because then we know, oh, there's somebody really good who would like to do this kind of bioinformatics research. Great, now we can go across the lab, work with our network and understand, oh, so-and-so wants that kind of similar expertise. And that's what drives the creation of an opportunity. So please reach out, even if you don't see something that immediately is an opening for somebody with your specific skill set. All right, and with this, I come to the penultimate section. I'm just going to share that my career has taken a very winding, non-predictable route. I did not know that I would wind up in a national lab. I did not know that I would wind up getting a PhD. I thought I would just get a degree in computer science, go off to some sort of startup and write code for a living. I kind of did that in my summer internships, but then it was the .com era. I was selling clothing online, which was a sort of an odd thing to do. And then I went on to grad school because the .com era ended with a whole lot of companies going out of business, including my startup. But ultimately, I wound up in numerical methods and high performance computing in the computational chemistry, biochemistry space. And that opened my eyes to the DOE through a DOE funded graduate fellowship, the CSGF, which led me to spend a summer at Argonne National Lab in just outside Chicago. I had a network there, and they knew roughly what I was interested in, and they said, "Hey, you should. I encourage you to apply for this fellowship position." So that was my first role out of a PhD. Not something I would have predicted, but it was fun. I was an outsider. To them, I was a biologist, even though sensibly very few biologists consider me a biologist. And I had two mathematical modeling problems that I really wanted to solve. I made significant progress on those. But at the end of my 2-year fellowship, it was the 2008 financial crisis. And those of you who are in graduate school right now might not remember much about this, but many universities canceled their hiring plans. So instead of going off to a faculty role, things fell through, and I went to work at the advanced in the biosciences division at Argonne Lab, specifically working with teams who are involved in the advanced photon source, which is shown here. Now that is a very, very cool facility because you have all these serious scientists riding tricycles around the very large ring, because you do not want them riding bicycles, because that is not a stable enough venue for you to transport your very, very priceless samples a long distance. So grown-ups riding tricycles, it happens. All right, so I had a mentor and collaborator who had 1 year of funding, and then I would have to find my way. All right, I'm grateful. Lee and I are still very close friends, and it turned out great. I proposed an absolutely outlandish idea, recruited some people who had expertise I didn't, namely in molecular dynamics, and it worked. It remarkably worked well to predict some of the things that we were taking at the advanced photon source. And I changed jobs in part because I did not succeed in the things that were expected if I had wanted to stay. So I want to provide that as a brief piece of career advice, is that if you go to a place and you'd like to stay, learn actively what is required and do it. I didn't, and that was my bad. All right, so 2009, I took a position in the Department of Biophysics and Physiology at Rush Medical Center, just outside Chicago. So you can see downtown Chicago up there, and way down here in the bottom right, not this fancy new building, but the one sort of right next to it in the edge of the picture, that top floor right at the edge was my office. Very cool, very wonderful place. But as you can imagine, an electrical engineer who does applied math and high performance computing, not a natural fit. It turned out that I had a bunch of constraints that made it impossible for me to take the faculty position I was offered. And that was okay. But I had a senior colleague who needed somebody to come in and sit on a relatively important committee at the hospital, the patient safety committee. And what they wanted was somebody who was willing to sit down, be very serious about listening, thinking things through, and asking questions. It was a really remarkably positive experience, and that was the basis for my receiving a faculty position. And I had mentors who explained to me, this is a medical school and you will not get tenure. All right, that's fine. But I did learn a ton, and it was great, great fun. To be in an experimental department and appreciate the reasons for their skepticism about some of the computing things that were emerging at the time. But as my mentors had said, that was never going to last. So after 3 years, I went from being a tenure track professor to a research assistant professor at Northeastern, which is a place that I hold in very high esteem, thanks to some great mentors. Basically, medical schools can be pretty dynamic environments, and high-level reorganizational decisions meant that my position was eliminated. Nothing personal, good relationships with everybody, and I just needed to line up 2 jobs, at least a couple, hopefully quickly. I chose to go to Northeastern, in part because I expected that was the end of my academic career. I would wind down my research, get those last few papers out, and then switch to industry, where I had a network in Boston, due to where I went to college. But as it turned out, one of my NIH applications was funded, so I got to stick around the research environment a little longer. Yet another weird winding turn. That NIH award plus another one from the Army let me actually convert to tenure track in the Department of Mechanical and Industrial Engineering. Now, I have no background in mechanical and industrial engineering, but I did have something really interesting, namely the mathematics between the models I was doing of proteins had a really beautiful connection to the models that some of my colleagues in that department were doing with nanomaterials. You think about a solution of proteins, like in a vaccine, for instance, an antibody vaccine, you got these things that aren't water, aren't the solution. And in nanomaterials, you have these defects, maybe they're carbon nanotubes or quantum dots or whatever, but you have these inclusions in the solid material. And it turns out the mathematics to describe the macroscopic properties of both have a deep connection. So the idea was that we would pursue that analogy. I was very grateful that they would take a chance on a non-mechanical engineer, and it was very fun. I had some great students and a fantastic postdoc in the mechanical engineering department. Folks I never would have gotten to work with without that kind of sort of unusual crossover. But after a few years, I decided I needed a change of pace. And so I went to GlaxoSmithKline. The industry environment, especially at a big pharma, is very, very different from the university structure and culture. I had a really strong mentor and leader and learned immense amounts from her. And I still reach out to her to understand, sort of what I'm like and how I might be reacting to things, as well as to understand the broader dynamics of the research world that she sees. I walked away from my academic research, not expecting ever to go back from the industry environment. I didn't get to go do my research expertise in industry. I hoped, but it didn't happen. But turns out there's lots of interesting problems out there if you're willing to be open to people saying, can you help? And if you say yes, lots of doors open. Over the next few years, I had 4 positions in under 3 years at GlaxoSmithKline. You might hear about pharma being a highly dynamic organizational environment, and that was great fun. I went into the global IT division to try and help some of their senior leaders understand what the research side thought was important in AI analytics and modeling, but ultimately came back to the research side to try and help R&D scientists connect to high-performance computing capabilities and build a partnership across different pharma companies to capitalize on the opportunities of quantum computing. But 2020 came around, and the DOE's mission in climate change and the environment really was, I couldn't resist coming to EMSL. I have a lot of longtime mentors who are at PNNL. I really just thought the chance to help grow this computing area at EMSL was totally unprecedented for me. And it was a chance for me to leverage some of my industry experience, some of my academic expertise, and most of all, my enthusiasm for working with a wide range of researchers to push their capabilities and their science agendas. All right, so I'll close. There's a bunch of resources that I wanted to add, whether that's on the web or Twitter or LinkedIn. Our social media platform is pretty active and a lot of fun. We also have a essentially monthly webinar series, which I encourage folks to have a look at. The previous ones are available on the YouTube channel. And for folks who want to understand the larger thrusts of the evolution of EMSL and DOE activities, each of the program offices has an advisory committee, which generally speaking, you add AC to the end to get the name of it. BER reports are available at one link, and the OSCE reports are available at another. They offer a pretty amazing set of insights into the evolution over the last 20 years of these very, very important funding agencies and mechanisms and their priorities. You can also reach us via email. The official EMSL email address is below, as is mine. And I thought some people might find specific books that I found interesting to sort of have thoughts on. And that I will make these slides available as soon as we are done here. But with that, I just wanted to say thank you and ask if there are questions.
All right, thanks so much for your talk. I will work to monitor any questions in the chat, or if people want to raise their hand and ask questions, they can do that as well, and I'll call on them. But while people are thinking of questions, I can start off with one. So I was curious if you could talk a little bit more in depth about like what a day-to-day work schedule looks like for you and your work at PNNL, and maybe also what that would look like if someone was to come, you know, out of a PhD program or end up in a scientist or postdoc position at PNNL in bioinformatics, what kind of work they would expect on a day-to-day there.
Okay, thank you. My day is pretty fragmented because the role that I have involves a combination of strategy development with leaders within EMSL as well as across the lab. So we're trying to, for instance, make sure that advances in AI that are made in another directorate are things that we can bring in and make available to the BER community as part of our user program. So trying to keep all that in sync has one kind of cadence. I support a number of the user projects that are in the computing space, and the granularity of that work could be anything from helping make sure that a particular piece of software runs at all on Tahoma to consulting with them to discuss what, you know, kind of pattern would you like to come back? Would you like us to come back to you in a month or 2 months? Or, oh, there's this new piece of software. Should we get a license to it? Would that help you out? So there's a sort of direct hands-on management of user projects. I also am a thrust lead for one of the lab's initiatives. As the lab stewards a number of 5-year initiatives, which are intended to be strategic investments in particular areas. For instance, this one is called predictive phenomics. And the idea is that we're going to develop new bridges between our biological modelers, people who do metabolic modeling, people who extract understanding from biological sequence databases, and so forth, and connect them to some of the novel experimental modalities that people are bringing to bear, you know, in the wet labs. So we are stewarding, you know, right now I think it's about 8 projects, and next year it'll be 12 projects, 4 new ones. And so there's a whole rigmarole of stewarding that scale of a research program. That's great fun because I'm working with scientists as well as connecting with the high level lab leadership as to where they want the initiative to go. And I wear a hat for the OSCAR, the scientific computing side of DOE, which is to help connect researchers who, for instance, are climate modelers who need to work with applied mathematicians from that part of DOE Office of Science. And we work in a computational partnership. So I help be their point of contact to the Office of Science in that respect. For folks who are just joining straight out of graduate school, you might be split across 3 or 4 projects. So the mix might vary on any given time of year. You know, you might spend some months, it might be 50% on one project and just a little bit on the others. Other times of the year it might be pretty evenly split depending on conference deadlines and travel and so forth. But in general, one would expect to be split across a bunch of activities, and you work with your line manager to find the right balance so you're not doing too many things and feeling spread too thin. I see a question in the chat. Do most positions at national labs require US citizenship? Not all do, many do not. There's extra work to do if one is not a US citizenship, but for most positions, I would say it is not required that you be a US citizen. Other questions?
Hello, can I ask a question?
Please.
Hi, Jay. Oh, thanks for the great talk. It contains a lot of important information. So I am an applied mathematician. I do PD modeling, and so far, the grants I have been applied has been only NSF because DOE grants usually they are very big, and in order to get this big grant, I need to seek collaborators. Well, I guess interdisciplinary, right?
Yes.
And so I have been having like trouble to find collaborations with people from industry or even other fields in STEM. So do you have any suggestions on like how should I find collaborators from other fields in order to get grants in DOE?
I think thank you for that question. It highlights something that is really distinctive about DOE science, which you put brilliantly, which is that DOE projects tend to be quite large and highly interdisciplinary. And so as a result, having a team that is ready to go when a call comes out is often really, really important. I think one thing that I would recommend, at least on the DOE side, that's all I'm going to restrict my answer to that scale, is to reach out to folks at the different labs who you see publishing, and just get to know folks. We're very happy to have people in, whether virtual visits are now super easy to do, or in-person visits as those return. To meet a wide variety of folks, whether in the same area of expertise and approaches, or their collaborators to build new connections. I think that would probably be my first recommendation. But please feel free to email me and say like I'm interested in meeting these folks, or this is where I'd like to be going, because it is hard to build those connections for a DOE scale award in the absence of ongoing collaborations. But we have to start somewhere. I just start with like, let's find some visits to the different labs or other institutions.
Okay, well, thank you so much.
You're very welcome.
Okay, so we have a question from the room. Do you want to say your question aloud? I'll be able to hear you. Do you want to state your question?
Yeah, just general question regarding like the living costs around the national lab there, as well as like what's the compensation outlook?
Cost of living in Richland is fairly low compared to major urban areas. One of the significant factors transitions that every employer has made over the last few years has been the rise of a lot more remote work. I don't live in Richland, Washington. I am calling you all from my house in Philadelphia, and I have no plans to move to Richland. And the lab and Battelle, which manages the lab, is very progressive in recognizing that for any of a number of reasons, potential employee may be unable or unwilling to move to the certain locations. There are certain job functions. If one is a wet lab scientist, it might be hard to do the job from another state. But many roles are it's much more possible and tolerable and embraced to have strong people working wherever is best for them and their families, as opposed to having to move to the sites that were established 50, 60, 70 years ago for other reasons. DOE is aware that the laboratories can be in relatively remote locations, and that may not work for some families. So as far as compensation goes, I'm not sure exactly what to say. It depends so much on your particular field and expertise. The labs and the DOE system are working very hard to ensure that very talented people choose to join the labs and stay at the labs. And I will say that I did not take a hit to my salary when I joined the lab from industry, which is saying something. Does that answer your question?
You said yes. We also have another chat question from Calvin Luther asking which programming languages dominate for models run on your high performance computers, and is it common that you would need to rewrite into a suitable language?
Oh, an excellent question. I would say that what currently runs on our HPC systems is dominated by C, C++, and Fortran. I presume that's not too much of a surprise. Increasingly, we do see a lot of folks running R analyses in R or in Python. We have a lot of excitement around Julia for a variety of reasons. And as far as I can tell, the future seems to hold a lot of interesting opportunities at that intersection where we spent many years with the Intel kind of processor stack, and it's taken us 2 decades, a little bit more, to get GPUs even approximately included as part of the mainstream. But over the next 10 years, the diversity of computing architectures and accelerators is going to multiply. It's going to be spectacularly different. And so the goal is one of the opportunities is to identify those key languages or libraries or programming models where, as that diversity of computing architectures increases, we're not going to have to do the substantial rewrites that we have had to do over the course of the last few decades, whether that was the craze or now with GPUs. Gosh only knows what's going to happen with quantum. But we at EMSL are not doing a whole lot of rewriting into suitable languages, although there are key parts of some codes where we are looking at very seriously, are our mission needs served by undertaking and investing in that kind of rewriting. But in general, it's not something that we do. We want to help people catalyze doing that or recognizing that doing that would be valuable. FPGAs are another great example. We do not currently, as part of the user program at EMSL, but we do have a very, very substantial team that is using FPGAs in reconfigurable computing and the software stacks associated with FPGA accelerated computing. Kelvin, does that answer your question?
All right, everyone. Well, thank you so much for joining us today, and thanks to everyone for coming to this seminar. I think if that's the last of the questions, we can wrap it up for today. And if anyone will make the slides available later, and of course, it seems like Dr. Bardhan is willing, if you'd like to reach out to him for more information on anything that was presented in this talk, then you can do so. All right, so thanks everyone, and enjoy the rest of your day.
Thank you very much, everyone. Be well.